Next, I'm going to discuss our Maxam Gilbert sequencing method. Maxam Gilbert method sequencing is method of DNA sequencing. It developed by Alan Maxam and Walter Gilbert in 1976 to 1977. Maxam Gilbert sequencing was the first widely adopted method for DNA sequencing, and along with the Sanger dideoxy method. This method is based on chemical modification of DNA and subsequent cleavage at specific nitrogen bases. We can cut the nitrogen bases. The principle of Maxam Gilbert method basically is purification of DNA segment that to be sequenced and labeled with radioactive material. So secondly, the chemical treatment generates breaks at specific nitrogen bases, and thus a series of label fragments is generated. We can generate label fragments. The fragment in the four reactions are arranged side by side in gel electrophoresis by size separations. They can be separated according to their size. Their two size, small or large. The fragments visualized in X-rays for autoradiography. To visualize the fragment, the gel is exposed to X-rays film for autoradiography, yielding a series of dark bands, each corresponding to a radio-labeled DNA fragment, from which the sequence may be inferred. As you can see in the diagram, sequencing an oligonucleotide by Maxam Gilbert method. So we can have a sample DNA and we can prepare of homogeneous single stranded DNA. So that is the sequence of DNA. We can add up of 32p as 5-phosphate. In position 5 phosphate we can add p32. So these are the sequence, and we can cut it at specific nucleotides. So if we can cut it, there are different reactions are formed. If we can cleave it, there are 4 different reactions are formed. G reactions, A reaction with some G cleavage, T reaction with some C cleavage, C reaction. So these are the sequence of these reactions. After cleavage, we can undergo the process of electrophoresis. So we can see it after electrophoresis, we can move on process of radioautography, though the fragment can be aligned according to length. These are the whole oligonucleotides, and these are the read sequence. So that is the process of sequencing an oligonucleotide by Maxam Gilbert method. Procedure. Maxam Gilbert sequencing require radioactive labeling at 1/5 of DNA fragment to be sequenced. That is gamma 32p. We can already discuss in previous diagram. The addition of 32p at 5-phosphate. So the chemical treatment generate break at small proportion of 1 or 2 of the 4 nucleotide bases in each of 4 reactions. These are guanine, adenine plus guanine, cytosine plus thymine. For example, there are different example. The purines A plus G by using formic acid, adenine plus guanine, that is purines. The guanines and the guanines, and to some extent the adenines by dimethyl sulfate. The pyrimidines, cytosine plus thymine is the pyrimidine by using hydrazine. Sodium chloride, that is NaCl add to hydrazine for cytosine. So in next process, add each chemical in a separate tube. Thus, a series of label fragments is generated. The fragments in four reactions are electrophoresis side by side for size separations according to their size. We are already discussed or small size and large size. So we can electrophorese it. To visualize the fragment, the gel is exposed to X-ray film for audio radiography, yielding a series of dark bands, each showing the location of identical radio-labeled DNA molecules. That is the procedure of Maxam Gilbert method.